Hi guys, first time I've done this, so bear with me. We're on the heart and the blood vessels. Some of you have asked me to go through this, so here we go. So firstly, the heart and the blood vessels in humans is in what's called a closed circulatory system, and that means that blood is always in the blood vessels. It doesn't leave there. It moves just in the tubes, delivers to the organs, capillaries pick it up, bring it back to the veins and bring it back to the heart again. Whereas in some smaller animals, you'd find an open circulatory system, and that means that the blood just flows around kind of loosely. Diffusion is a movement method, and diffusion is the way that small animals kind of transport their blood around the body. Okay, so that's the first thing. So arteries have thick walls. They carry blood away from the heart. A for arteries, A for away. Because they have a narrow lumen and they have thick walls, that means the blood is under really high pressure, and it's on the arteries that we get our pulse because of that outward pressure. Arteries carry oxygen-rich blood except for the pulmonary artery, and you'll see why later. So sometimes you're asked to draw a diagram of what a cross-section of an artery looks like and then a cross-section of a vein. So this is what you kind of need to draw here. So the vein will have a big lumen, so the space in here. You can also add in some valves in there as well, something along those lines. Because veins have valves, again, V for vein, V for valve, and they also have a thinner wall here. Compared to the artery over here, they have really thick walls. Like I said, the smaller lumen, that means the blood flows very, very fastly. On the outside of both, there's inelastic collagen, which keeps its structure. And you can tell that yourself. Sometimes if you press in on the veins on your hands, they kind of spring back out. To their normal shape, really. Okay, so that's the structure of arteries and veins. Capillaries, really, really small, only one cell thick, and that's a feature that allows them to allow particles to come in and out. And where the cells meet, you see these pores. That's where some food can kind of go in there and be picked up and be transported through the closed circulatory system. So veins carry blood away from the heart. Just go in here. Sorry, wrong, my mistake. Veins carry blood to the heart. Arteries are away. They have low pressure. Veins will have thin walls. They have valves, which is another difference, a structural difference between arteries and veins, and they have large lumens. Okay, so I talked about capillaries. They connect arteries to veins and have thin walls. Valves prevent the backflow of blood, and sometimes you're asked how valves allow blood to kind of get back to the heart. So here we have a person's foot, and while sitting, the blood can gather down in these areas. So how, against gravity, can the blood get all the way back up towards the heart? Well, the skeletal muscles in your body contract, and even if you're inactive, it's good to stretch, because when you stretch, you squeeze the muscles. These muscles squeeze in, make the vein a little bit smaller, the lumen a bit smaller, and that pushes the blood up a little bit. And that force there, as you can see on this person's right leg, that squeeze pushes the blood up, pushes it through the valves, and then the blood, if it tries to fall back down due to gravity, will close the valves like this one here. So then the only place for the blood to go is upwards, and eventually it'll get back into the heart. Okay, so the blood flow through the heart is a double circulation. That means that it goes from the heart to the lungs and then back to the heart. That's the pulmonary circuit. Then it goes from the heart to the rest of the body and back to the heart again. So this is a diagram here of the heart. You do need to be able to draw this. I'll show you how to draw it now kind of later. But you need to know all of those labels there. Okay. So the heartbeat is controlled by the SA node, which is in the wall of the right atrium. So you see it there in the diagram. And the atrioventricular node, which sends the electrical impulses down the septum to the ventricle. So the the SA node is up here, and then the AV node is in there. So this is a lot of nerves that will extend all the way up to the brain. Each time your heart beats, the nerves send an impulse down to the SA node that contracts, which causes the blood in the right atrium and the left atrium to squeeze and contract and push down to the ventricles. Once it's down in here, there's a slight delay, and then the AV node will pick up an impulse from the brain, which will cause the ventricles to contract, squeezing the blood out of the right atrium up the pulmonary artery, and from the left ventricle, ventricle up here through the aorta up to the rest of the body. Alright, so that's how blood flows around. So the two stages are diastole and systole. And diastole means when the heart is relaxed, and systole means when the heart contracts. So we had an experiment that we did where we investigated the exercise on a pulse rate of a human. So we got our pulse rate at resting, which was the control, straight after exercise, 1 minute after exercise, 2, 3, 4, and 5 minutes after exercise. What you would likely to see is the average BPM, which is around about 72 beats per minute. Straight after exercise, you might get all the way up to 120, depending on how fit you are. 1 minute after exercise, you would expect it to decrease. 2 minutes after exercise, again, probably decrease or around the same, depending on your fitness levels. And eventually, it should fall maybe even below your resting, or somewhere around that. It might just be exact bang on. The reason why it goes below, like it did here in this person, is because the body senses that enough blood has gone around the body to deliver oxygen, which is carried in the red blood cells, to the muscles that had been working. So then the brain says, okay, hold on, we've picked up enough oxygen, so let's slow down the beat. And that's why sometimes you can get down to 70. Okay, so blood pressure is the force of blood against the walls of the arteries. Normally the higher value is a measure of the systolic pressure, and the lower value is a measure of the diastolic pressure. Can you remember what it is? I'll give you a clue. The unit is in mmHg. So the higher value, which is the systolic pressure, which is when the heart is beating, is 120 usually. The diastolic pressure is when the heart isn't beating. That is usually 80. Okay, and that's something that you need to know there. 120 over 80 millimeters of mercury, Hg. Alright, okay, so smoking is very damaging to the heart because it increases heart rate, increases blood pressure, increases the risk of blood clots. There's the ingredients of a cigarette. If you're trying to convince anyone to stop, please do. Also, the main dietary factors affecting the circulatory system are saturated fats, which cause arteries to be blocked. So you get them in foods like chips, crisps, all the good things. High salt intake raises your blood pressure. Being greatly overweight raises blood pressure and causes heart attacks. Whereas exercise will strengthen the heart, because the more you use it, the better it gets, the stronger it gets. Like any muscle in the body, your blood circulation improves and your body weight reduces. Okay, so I've kind of gone through the stages of the heart earlier on, so I'm going to move on here. Okay, so a couple of questions here. These are just two that I'll post onto Edmodo as well, that you can try and write in as well, as long as a few others. The first one is, just let me expand it here, we'll do it together. So give one example of a limitation of a scientific method, just some revision. So this is accidental discoveries, changes in weather conditions, things like that. Where do scientists normally publish the results and conclusions of their investigations? That is in journals. Okay, so when dissecting a mammalian heart, we use the sheep. Precisely, where did you locate the tricuspid valve? Okay, we'll have a look at that in a second. Briefly describe how you carried out the dissection. Where did you find the semilunar valves? And when you investigated the effect of exercise on the human pulse rate or breathing rate, what did you first establish? So as I mentioned there before, that is the resting heart rate. How did you measure the pulse rate or breathing rate? We measured it by placing 2 fingers on our wrist or on our neck where we could feel a pulse. We timed ourselves for 60 seconds and wrote down our answer. Okay, make sure you include time there. Okay, so just going back to the experiment, how we identified the different parts of the heart. So firstly, what we did was we decided which side of the heart was which, and we did that because the left side of the heart is thicker, it is a thicker wall, right side is a thin wall. Also along the front of the heart, it's not in this picture because it's a kind of a cross section. Along the front you have a coronary artery that kind of branches off like that. Alright, so that's another way that you can identify the front of the heart compared to the back. So the question there asked was, how did you identify the different parts? So one of the questions was, how did you identify the semilunar valve? So there's a semilunar valve here on the way out of the pulmonary artery. So what you did was you dissected through the lunar valves in the pulmonary artery, and that's how you identified it. Another way is locate the aorta, dissect through there, and expose the semilunar valves there. Okay, so that's another way that you can identify that bit. Another question they asked was the tricuspid valve. How do you identify that? They can ask tricuspid or bicuspid. So in this case, I think it was tricuspid. So what you did was you identified the right side of the heart first, because it's the opposite of the left side with the thick wall. You placed a scalpel in towards the right atrium in here, and you made a cut down to the right ventricle like that. Okay, and you would have pulled each part to the side, and you would have found the tricuspid valve there right in the middle, separating the right atrium and the right ventricle. Okay, so one question did appear recently. I think I have it here. No, that's the question from before. Yeah, okay, so this one. In the space below, draw and label sufficient of your dissection to show the tricuspid valve in the right atrium and the right ventricle. So they're asking you basically to draw the heart or a section of the heart. It's quite difficult to do just that small section, so it's easier just to draw the whole thing, and I'll just show you how to do it now. So what you need to do is draw the 2 tubes leaving the heart here first, close it off, slight bend, another tube out here, another slight bend, another tube out here, and close it off here. Okay, not the best start, but we'll go with this. So what you then need to do is label the tubes. So this one here is the vena cava, just VC for short. You have to do the whole thing. This one over here comes down from the lungs, and that's pulmonary vein because it's coming back into the heart, so it must be a vein. Same thing over here with the vena cava, vein for vena cava. This tube here, just use a different color, is the pulmonary artery, and this one over here is the aorta. Okay, so what you need to do then is you need to draw a septum down the middle and label that. Okay, so then what you need to do is on the right side, which is this one over here, if you put in valves, and it's the right side, so it's called the tricuspid valve. Okay, those 2 letters there, RI for the right side. Then what you need to do is you need to separate the chambers. So I just draw a line down to there, and I'd label this the right atrium, and basically what happens is the blood will come in the vena cava. Down here the pacemaker will contract, push the blood through the tricuspid valve down into the right ventricle. Then our AV node, which is up here, which you can label in the wall of the septum, AV node will squeeze, causing the blood up and out the the pulmonary artery to the lungs, pick up oxygen, come back in the pulmonary vein, down in here, past the bicuspid valve, down into the left ventricle. The left ventricle then will collect it, and at the same time the right and left ventricles will be squeezing because of the AV node contracting, push the blood up and out of the aorta, branches off to the rest of the body. Okay, so just to finish off the labelling then, this one here is the bicuspid valve, and it's on the left side. Okay, so fairly terrible diagram there, but they did ask if we go back to draw the tricuspid, which I labelled, the right atrium and the right ventricle, which I also labelled there. So I would have got my full marks. So this is only a section, but make sure that you can draw the whole lot of the heart. Okay, so to finish off, underneath this post I'll attach some questions which are like the ones that I've screenshotted here. So if you want to give those a go as some practice. Okay, thanks very much.